Hi, my name is Michael Alden. I'm a Ph.D. student here at Clemson University, conducting research on cannabis with Dr. Jim Faust. One of the topics that we've looked at pretty extensively is photoperiod, and so that's what I'll be presenting today. So, during controlled environment production of cannabis, 2 photoperiods are typically provided, 18 hours followed by 12 hours. 18 hours is provided to maintain vegetative growth, and this would include things like maintaining stock plants or cutting propagation. And 12 hours is almost exclusively provided for flowering. So going into this research, we really wanted to evaluate whether these photoperiods are optimal, or maybe there might be some benefit from using photoperiods that are longer than 18 hours for vegetative growth, or maybe there would be some benefit from using photoperiods that are longer or shorter than 12 hours for flowering. In 2019, a study was published largely looking at the morphology of cannabis under long day and short day photoperiods. What they found was that solitary flowers would emerge even under long day photoperiods. So, because of this, they concluded that flower initiation in cannabis is a day-neutral response. However, development into the compact inflorescences requires short day photoperiods. So, from this study alone, we were just curious whether they were seeing these solitary flowers emerge because they were not providing a long enough photoperiod. Industry standard is typically 18 hours for maintaining vegetative growth, whereas in this study they provide only 16 hours photoperiod. Furthermore, we wanted to evaluate the range of flowering responses at different short day photoperiods. This study did not evaluate whether a 10-hour photoperiod, 11-hour photoperiod, or maybe even a 13-hour photoperiod would perform. So for this experiment, we used 3 different cultivars, Sunset, OG, and Tiger. And we know that these 3 cultivars have very different photoperiod responses. Tiger is a very early flowering variety, OG is kind of more in the middle, and Sunset is a very late flowering variety. 10 photoperiods were used in this study, 10, 11, 12, 13, 14. Those were kind of the range of the flowering photoperiods. And then we also provided 16, 18, 20, 22, and 24-hour photoperiods. At the beginning of the experiment, all plants were pinched and thinned to 3 shoots. And then 1 shoot from each plant was harvested after 4, 6, and 8 weeks of photoperiod treatments. So the entire experiment went for 8 weeks. Each plant had 3 shoots at the beginning of the experiment. And then 1 shoot was harvested on week 4, 1 shoot on week 6, and then 1 shoot on week 8. Now, diving into the materials and methods a little bit further here, what we're seeing in this photo is one of our greenhouses, and this greenhouse is equipped with automatic black cloth. The black cloth opens at 8 a.m. and closes at 6 p.m. each day. This provides a natural day 10-hour photoperiod. During this time, the plants are receiving sunlight as well as supplemental lighting from the LED lamps that are hanging above the plants. There are 4 benches in this greenhouse, and each bench has been subdivided with this panda plastic or these white plastic sheets hanging in between each zone, and each zone has 3 LED lamps in it. So all plants receive the same amount of light each day from natural sunlight and the LED lamps. However, to control the photoperiod, what we did was hang small LED bulbs within the individual zones, and these bulbs only provided about 2 micromoles. Before 6 p.m. each day, we would pull black sheets in between the benches, so that way we could prevent light pollution between benches. And then, of course, panda plastic prevents light pollution within each bench. All of the bulbs would turn on at 5:30 p.m. each day. And then the bulbs would stay on corresponding to the photoperiod. So at 6 p.m., the bulbs in the 10-hour photoperiod would turn off. At 7 p.m., the bulbs in the 11-hour photoperiod would turn off. At 8 p.m., the bulbs in the 12-hour photoperiod would turn off, and so on and so on. So overall, for this experiment, all the plants effectively had the same DLI, and what we're seeing as a result of this experiment are strictly photoperiod responses. Now, one parameter we collected data on for all plants during week 4, 6, and 8 was leaf number. Now, leaf number we counted as any leaf attached to the primary axis of the shoot with a petiole longer than 2 centimeters. So now what we see on this picture are these squares highlighting each node along this shoot. And so what we see is 1, 2, 3, 4, 5, 6 orange boxes, followed by the 7th box, which is in red. This is the last leaf with a petiole longer than 2 centimeters. What we'd expect from this experiment would be that flowering plants would have very low leaf numbers because all their growth transitioned to reproductive growth, and so no more leaves would be accumulated. However, we would expect that the long day photoperiods indefinitely produce leaves and internodes during the course of the experiment, so they should have very high leaf numbers. So, getting into the results of this experiment, the first variety we're going to look at here is sunset. In this figure, we have photoperiod on our x-axis going from 10 to 24 hours, and we have leaf number on our y-axis. We have 3 different trend lines here, each correlating to week 4, week 6, and week 8 of the experiment when we collect the data. Now, what's pretty interesting about this is that we have little to no growth that occurs in the 10-hour photoperiod. During week 4, leaf number is about 12.5, and then at week 6 and week 8, leaf number is right about 15. What we see in all photoperiods longer than 10 hours is we do see gaps in between the leaf number at week 4, week 6, and week 8. This indicates really nicely that the plant is still growing at each of these photoperiods. We see less growth at the 11-hour photoperiod compared to the 12 through 24-hour photoperiod, so this might be more of an intermediate photoperiod. However, from photoperiods 12 through 24, especially at week 8, the leaf number is all but the same. So, based on leaf number alone, you would assume from this photo that any photoperiod from 12 to 24 is equally effective at maintaining vegetative growth for sunset. Now, what's interesting about sunset as well, that this is a highly unusual response not to be flowering fully at 12 hours. What we see here are the 10, 11, and 12-hour photoperiods left to right 3 weeks following the experiment. What we found was that the 10 and 11-hour photoperiods would actually stretch pretty dramatically early on during the experiment. However, you can see that the 12-hour photoperiod is more compact and short. Furthermore, you can see in the 10 and 11-hour photoperiods that you actually have flower mass developing at each of the nodes, whereas the 12-hour photoperiod looks almost entirely vegetative. We have found with sunset that stretching actually correlates really nicely with the highest intensity of flowering response here. Now, of course, this stretching is not ideal, so other methods to control size can be used, such as pinching after the start of short days. Moving on to OG here, what we're seeing is the same structure of the figure here as we saw with sunset, where we're seeing photoperiod on the x-axis, leaf number on the y-axis, and weeks 4, 6, and 8 as trend lines here. Now we see really no growth or no accumulation of leaves in the 10, 11, and 12-hour photoperiods from week 4 through 8 during this experiment. This indicates from week 4 on that the plants were completely dedicated to reproductive growth as no further leaves were being accumulated during this time. In contrast, beginning at the 13-hour photoperiod, we do start to see gaps in between week 4, week 6, and week 8. This indicates that the plants in this photoperiod treatment were still growing vegetative to some degree. However, the leaf number was lower than what they would be at, say, 18 or 20 hours. And so we know we have an intermediate response here. Now, what was interesting specifically with the 14-hour photoperiod here is that if we were trying to pick the best photoperiod to put plants under, we might assume that the 14-hour photoperiod is the best because it has the highest leaf number. However, what we found was that the leaves were heavily reduced in size. So even though we moved up the stem and we had petioles that were longer than 2 centimeters all the way up, we would really end up with leaves that only had 1 leaflet or 1 digit on them. In contrast, photoperiods from 16 to 24 hours had fully intact leaves. So leaf number is not the only metric we need to look at when we decide what photoperiod is optimal for vegetative responses. Now what we're looking at finally here is tiger for leaf number. And again, same figure, photoperiod on the x-axis, leaf number on the y-axis. And so what we're seeing here for the flowering photoperiods from 10 to 13 hours, really we're not seeing any growth here again. At 14 hours, we start to see just a tiny bit of growth week to week from week 4 to 6 to 8. And then We really start to see those first jumps at the 16-hour photoperiod from week 4 to 6 to 8. And then 18 to 24, we really don't see any differences in how much they're growing across these photoperiods. So as we see in the last 3 cultivars, from Sunset, OG, and Tiger, there are different ranges of where these photoperiods could be optimized just based off leaf number. And we would pick these photoperiods based on the lack of growth or the lack of accumulation in leaves across these weeks. We know with Sunset that the optimum photoperiod is really only 10 hours, so it doesn't make sense to really explore anything further than that. Whereas with OG and Tiger, there are a couple of photoperiods that do not really affect leaf number, and so that there potentially could be other things that could be optimized within each photoperiod differently. Now, jumping back to one of the first questions we had in this experiment was, could we prevent solitary flowers by providing a long enough photoperiod? And what we're looking at this photo is Sunset with the 18, 20, 22, and 24-hour photoperiods, but also OG responded exactly the same. So really this photo is the same for both cultivars. And so what we found was that at all of these photoperiods that there were solitary flowers. So no matter what photoperiod we provided, we could not stop the initiation and development of these solitary flowers even under long days. However, we found with Tiger that there would be solitary flowers on 18 hours, but 20, 22, and 24 would not have solitary flowers. So it seems at least the inhibition of solitary flowers under long days is cultivar variety specific, and this may just be a genetic component. So now moving on to flower dry weight, what we'll show you is the data we collected at week 8 during the experiment for both reps. So on the x-axis, we have photoperiod going from 10 to 14 hours, and on the y-axis, we have flower dry weight in grams per shoot. Now, if we remember back to how we conducted this experiment, we only had 1 shoot per plant left by week 8, so the flower dry weight here is representative of the entire shoot. So starting with Sunset, what we see is a slightly lower flower dry weight at 10 hours than we do at 11 and 12 hours. Now this is pretty interesting because based off leaf number, we saw that really 10 hours was the optimum photoperiod for flowering for Sunset. However, we're not seeing the largest amount of flower mass here that we would expect. What's also important to consider is how mature that flower mass is by week 8 of this experiment. So what we're looking at here is a photo of photoperiod treatments of Sunset at week 6. I trimmed up these plants to help make the distinction between treatments a little easier to see. And so what we're seeing is the 10, 11, 12, 13, and 14-hour photoperiods going from left to right. And what you can see pretty clearly is that the 10-hour photoperiod is actually developing quite a bit of flower mass, and you have less and less flower mass developing at the 11 and then 12-hour photoperiods. So you can see the strength of the photoperiod response alone just between the 10, 11, and 12-hour photoperiods. And then at 13 hours, and especially 14 hours, these plants are almost entirely vegetative again. So now what we're looking at here is Sunset at week 8, only looking at the 10, 11, and 12-hour photoperiods. Now, if we look at either of these photos, because the right photo is just a blown up version of the left photo, what we see here really nicely is that we actually have the densest flowers in the 10-hour photoperiod, and we have less and less development as we go to 11 and 12 hours. Furthermore, we only really have these orange and brown stigmas in the 10-hour photoperiod, and we don't have that in the 11 or 12. This would suggest that the 11 and 12-hour photoperiods are less mature than the 10-hour photoperiod. Now, based off this photo alone, you would figure that the 10-hour photoperiod would have more flower mass than the 11 and 12. However, this is only the top 20 centimeters of each shoot from these treatments. And so the 10-hour photoperiod is fairly short, whereas the 11 and 12-hour photoperiods are very long shoots, and as well as the laterals that are on those individual shoots have a lot more flower mass overall than the 10-hour photoperiod. Jumping back to our flower dry weight, now we'll look at OG, and we see that the 10 and 11-hour photoperiods for OG are fairly similar. We start to see a small increase at 12 hours, but not a significant difference. And then we start to see a large increase at 13 hours for flower dry weight. So we have OG here at week 6, and photoperiod again going from 10, 11, 12, and 13 left to right. And so really, we don't see any differences here between photoperiods 10, 11, and 12 for OG. However, we do start to see the 13-hour photoperiod that the flowers are smaller. We don't see the dense accumulation. We see less trichomes or less frostiness on these bract leaves as well. And now these are the same photoperiods of OG at week 8. And so similar to sunset, we actually now have 2 photoperiods with brown and mature stigmas. At the 10 and 11-hour photoperiods, we see very dense accumulation of flower, as well as brown and orange stigmas predominantly. In contrast, the 12-hour photoperiod is predominantly white stigmas, indicating that these flowers are less mature than the 10 and 11-hour photoperiods. Then finally, at the 13-hour photoperiods, again, we see these very small flowers, much reduced in trichome density as well on the bract leaves. Now, keeping in mind from that last figure as well that we saw the flower weight was much higher at 13 hours than 10 through 12. But again, that's a little misleading from this photo alone because the shoots are so large in 13 hours compared to the 10 through 12-hour photoperiods that it does end up having more flower mass. Now, finally, looking at this figure for tiger, what we see are pretty much the same response for flower dry weight at the 10 and 11-hour photoperiod. At the 12-hour photoperiod, we definitely see an increase. And then at 13 and 14, these are pretty similar to each other. And so we have 10 and 11 pretty similar, 12 in the middle, and 13 and 14 pretty similar to each other. Now this is tiger at week 6 for photoperiods 10 through 14, left to right. And so we really see pretty similar responses with 10 and 11, and especially 12. They really don't look any different. However, you do start to see a little bit paling of the green and a little bit less density of the flowers with 13 and especially 14-hour photoperiods. And then finally, we have tiger at week 8. And so the photo on the left really is a nice representation of the photoperiod response of tiger because we see the shortest plants at 10 and 11 pretty much identical to each other. 13 and 14 are pretty much identical to each other, and 12 kind of sits nicely in between the two. Now, what's interesting and unique about tiger is that even out to a 16-hour photoperiod, we do see slight compaction of these lateral shoots, as well as a pretty decent amount of stigma accumulation, or these solitary flowers accumulating at these node positions, even on the lateral shoots. So for tiger especially, a 16-hour photoperiod would not be acceptable for maintaining vegetative growth, and at least an 18-hour photoperiod would need to be provided. So now after looking at flower weight, what we wanted to do was look at the cannabinoid content of the flower mass that we harvested. To do this, we harvested all the flower mass off of each shoot for the plant, and then we grounded up all the flower and then sampled 2 grams from that and sent that off to the lab. And this is the resulting figure. So again, we have photoperiod on the x-axis going from 10 to 14 hours and CBD percentage on the y-axis. And again, we have sunset, OG, and tiger. So with sunset, we do see a little bit lower of CBD percentage at the 10-hour photoperiod relative to the 11-hour photoperiod, but then we see a huge drop-off at 12 hours. Similarly, for OG, we see a low CBD percentage at 10, which increases at 11 and increases again at 12 hours, but then drops off dramatically at 13 hours. Finally, for tiger, we have a low CBD percentage at 10, increases at 11, increase at 12, increase at 13, and then finally drops off at 14. So what we see here are different optimums based on CBD percentage for these different varieties, meaning that each variety potentially has a different photoperiod where optimal CBD percentage or accumulation and synthesis will occur. Finally, if we want to look at total harvested CBD, which is the interaction of flower dry weight times the CBD percentage, and that's what this figure looks like here. And so again, what we have on our x-axis is the photoperiod going from 10 to 14 hours, total harvested CBD in grams per shoot on our y-axis, and there are 3 cultivars here. And so this figure gives us a pretty similar idea as the last figure, being that there is one photoperiod that is really optimal for total harvested CBD. Now this figure gives us a pretty clear answer if we were growing strictly for extraction. Whereas if we only cared about the total harvested CBD per grams or per shoot, then an 11-hour photoperiod would be used for sunset, a 12-hour photoperiod would be used for OG, and a 13-hour photoperiod would be used for tiger, because these were the highest producers of CBD. However, what we found was more mature and typically denser inflorescences in photoperiods shorter than where CBD synthesis and accumulation is highest. So especially for OG and Tiger, we found much nicer, denser flower at the 10 and 11 hour photoperiods than we did at the 12 hour. So considering your end product is very important when it comes to deciding what photoperiods to flower your plants under. So one thing we've also looked at is the actual nutritional content of the flower in the different photoperiods for the three varieties. So what we're looking at is photoperiod on the x-axis going from 10 to 14, phosphorus in parts per million on the y-axis, and our three cultivars, Sunset, OG, and Tiger. Now what's pretty interesting is that obviously each of these cultivars has one or two photoperiods where the phosphorus levels are the highest, and then increasing photoperiod beyond that, where the phosphorus is the highest, leads to a decrease in phosphorus. So for example, with Sunset, we have our highest phosphorus levels at the 10-hour photoperiod, and if we go to 11 and 12, our phosphorus levels decrease. This is pretty similar to what we saw with leaf number, where we saw the lowest number of leaves in the most inductive flowering treatments. So we could really tie in nicely and look at the flowering intensity as a function of leaf number and phosphorus concentration. So overall, we can summarize the experiment pretty well with this one figure. What we have here is photoperiod going from 10 to 24 across the top, and Sunset, OG, and Tiger on the left. We've broken the different photoperiod responses that we've observed in this experiment into these different colors. When it comes to flowering, deciding what photoperiod to use depends on what your end goal is. Are you growing plants for smokable product, or are you growing plants for extraction? For Sunset, we obtained the most compact, mature flower only at the 10-hour photoperiod. This definitely had a lower yield overall, but these were much better flowers relative to any of the longer photoperiods. At the 11-hour photoperiod, we got higher flower mass yield as well as CBD percentage, leading to greater overall harvested CBD per plant or per shoot. However, this flower would not have been acceptable for smokable product, and this photoperiod would have been optimal for extraction. Then what we found at 12 hours was that this was more of a transition photoperiod where over the course of 8 weeks, these are really not acceptable for harvesting for smokable product and had much lower CBD percentage than the 11-hour photoperiod. Then what we found from 13 to 24 is that there are really no differences with these plants. And this trend holds true for OG and Tiger as well. With OG, we found almost identical compact mature inflorescences from 10 and 11 hours. We had a higher yield of CBD off the plant at the 12-hour photoperiod. And then 13 to 14, we had transition. And then 16 to 24, these plants were all effectively the same. Then finally with Tiger, we really had a pretty similar range of dense compact flower from 10 to 12 hours. We had our highest size and yield for extraction at 13 hours. 14 to 16 were more transition photoperiods, and then 18 to 24 were our vegetative treatments. Overall, 18 to 24 hours photoperiods were similarly effective in keeping plants vegetative, and there were really no differences in quality among these photoperiods. And we really found that variety evaluation would probably go a long way with deciding what photoperiod to flower some of these varieties out. Now, of course, there are limitations with grow areas where and how plants can be black clothed for different photoperiods. However, there are different optimals for different varieties, and that the photoperiod you use should be based off of whether you're growing the plants for smokable product or extraction. So future possibilities from this project could really be looking at providing different photoperiods during long days and short days. One such example would be looking at the cost or the efficiency of providing, let's say, anything from 18 to 24 hours. So for example, you could provide a lower PPFD at 24 hours and obtain the same DLI as you could with an 18-hour photoperiod that had a higher light intensity. So how the actual economics and the efficiency actually works on that should actually be evaluated. Another possibility, which we might be pursuing this fall, would be starting the plants under a 10 or 11-hour photoperiod for the first week or two, and then moving them to a longer photoperiod to finish. This ideally would result in compact inflorescences as the buds or the meristems transition to flowering, and we have really short internode spaces in all of the inflorescences. And then we go to a longer photoperiod to really maximize the DLI and thereby the yield. So that's all I have for you today. Thanks so much for listening. We'll see you here back next month. Thank you.